Okay, guys, so DNA structure and function, as well as replication, is going to be absolutely huge in the discovery of DNA's structure and basically how it goes about doing all of the amazing things we've learned about it doing. As we know, Watson and Crick were the ones to really put together the first model of DNA as far as the hydrogen bonding and everything goes. But let us not forget about Rosalind Franklin and her significant contribution to that with UV crystallography as well. So DNA structure and function, its structure allows it to perform its function of storing information in a way that can be copied and passed down to future generations. So in your notes packet, that's shown here. And it's really important that we understand that the very unique structure of deoxyribonucleic acid allows for it to be passed on to generation after generation. And this is how it does it. So the structure shown here, or complementary base pairs, this is basically telling us, only thymine can bind to adenine and only guanine can bind to cytosine. And of course, we can incorporate RNA in there. But this is the number of spots where hydrogen bonds can occur between the nitrogenous bases. So up here, I'm going to write on my notes packet, complementary base pairs. And this is the genetic message is conserved as a result of these complementary base pairs. The next part that we look at is the backbone, and this really works to maintain the order of bases. So the sugar phosphate backbone that we learned about, it's going to help maintain the order of the bases. And as we're going to see, and as we've seen a bit before, that order is essential. And so next part that we're going to look at here, the hydrogen bonds. These hydrogen bonds, as you'll see in the presentation there, these hydrogen bonds are pretty weak, right? We've learned that they're actually more like attractions than bonds. So they can break and reform with just a little bit of energy. And that's great in terms of DNA replication because an enzyme, aka helicase that we're going to learn about, can unzip and rezip the DNA easily during DNA replication. So this requires little energy, which remember, energy is very important, requires little energy for the bonds to be broken, allowing for easy replication, and don't forget transcription and translation as well. Actually, transcription. Mentioning replication, we need to understand that DNA replicates in a semi-conservative fashion. What this means is that the parent strands are broken apart and then they are used to read and use complementary base pairing to make a new strand. So each parent strand contains a new synthesis of a complementary strand. So in your notes, you should see a little diagram that looks like this. Semi-conservative shows that we are able to have an original strand paired with a new strand. Conservative is an alternative. However, conservative replication leaves the two original template DNA strands together in a double helix and produces a copy composed of two new strands containing all of the new DNA base pairs. I want you to think about why that might not work very well. What would be hard about that? And then the next alternative to semi-conservative is that of dispersive. So this is when DNA containing distinct regions of DNA composed of either both original strands or both new strands. So kind of getting this like random mishmash of different DNA, old and new. We have since supported the semi-conservative model. One thing I'd like you to try and do is to start with a single DNA double helix, then draw 2 cycles of DNA replication, drawing all new strands in a different color using the semi-conservative format, right? So if we were to do this, I'd like you to pause and try to do that in your notes. But if we were to do this together, it would look something like this. So I want you to think through, how could DNA scientists tag DNA with different colors to show this pattern? And this is actually what took place with Meselson and Stahl experiment in 1958. And the way that they did this was using E. coli, and they were able to replicate E. coli bacteria and then visualize just what happened. Long story short, they were able to incubate E. coli bacteria into a medium that had heavy nitrogen in it. And so now all of their new DNA, wherever nitrogen was, is going to have that marker of heavy nitrogen. They then went about separating it and getting all of the heavy DNA and only used that heavy DNA. And so I'm going to compare the 3 different types of replication. Cycle 1, the bacteria were transferred to a solution with light nitrogen and allowed to divide. And when we saw that, let's see, so for each one, draw the DNA and resulting DNA density for each proposed mechanism of DNA replication, as shown here. So if it were semi-conservative, we would see a heavy chain, the purple ones, paired with a light chain because they would have to pair together with those new nucleotides. With conservative, it would be all heavy and all light. With dispersive, it would be intermixed. But what we saw is that when spun with a centrifuge, all the DNA was in a single band above where the 15N band had been. What that tells us is that cannot be conservative then, because you would have had 2 separations. You would have had one heavy band below and one light band above. It couldn't have been that way. So the bacteria continued to grow with light nitrogen and allowed to divide. And so we know it can't be conservative. So with semi-conservative, what will we see here? Let's keep on moving through. We know that it can't be dispersive because we're getting this mixture. They're splitting into kind of 2 separate places. So ultimately, semi-conservative, is going to show that eventually all of the old heavy DNA is going to be left out and the new DNA is going to be the one that's mixed in. So when spun in a centrifuge, some DNA was in a band above where the 15N band has been, and more of the DNA was above that, showing that the old DNA that was replicated kind of got left behind gradually because those old strands were not used in those newer replication process. I've put here a link for you if you access the PowerPoint presentation in Google Drive, and you'll see a little bit more detail in a video of Meselson and Stahl's DNA replication. Now to actually learn how DNA replicates, we've done a bit of this in class. If you remember, we started this together, but the model we built was typically with our human bodies. And so, again, Watson and Crick's famous phrase, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." So if you can, try and remember and pause the video drawing a DNA nucleotide. Hey, guess what? I have a sugar on my shoulder, a phosphate on my fist, and a base on my hand. I am a nucleotide, right? Try and draw that out. Pause the video here. And remind yourselves, okay, I have a sugar. That's going to be my ribose or deoxyribose. I have a phosphate on my fist, right? PO4. And then I have a base on my hand. Remember that the phosphate on the fist represented that 5' end, okay? So if you can now draw another nucleotide and connect them covalently, you're going to start seeing that, okay, they attach together their 5' to 3' ends, right? So that 5' sugar was going to, or 5' phosphate end is going to bind to that 3' sugar shoulder, okay? Looking ahead, now if you can connect two additional nucleotides hydrogen bonded to your previous, try and think about what that might look like. And then notice here, remember we have a 5' end with that free phosphate standing out, a 3' end, which is a free hydroxyl, and then of course, so in starting our understanding of DNA replication, the first thing that we're going to have is a helicase that is going to break hydrogen bonds between bases, which unzips and unwinds the helix. So in my notes here, I would write breaks hydrogen bonds. Oops. And begins to unwind the DNA at the origin of replication. So it's common to only show one strand's DNA sequence since the other can be inferred. So oftentimes we're going to just show one strand. So up here, begins to unwind the DNA at the origin of replication. Helicase enzymes move in both directions from the point of origin, forming as you can see in the PowerPoint here, it's going to form a replication bubble.